So let's see where we left off. In the last unit of study, we had, in glycolysis, we had converted glucose into two pyruvate molecules. Now, pyruvate in this case was the waste product. If we're just looking at glycolysis, we had to go through fermentation to convert pyruvate to something else. We had to use fermentation to cycle NADH back to NAD so that glycolysis could continue. But we find now that the end product of glycolysis is actually the starting material for this next energy production pathway, and that would be the citric acid cycle. Ultimately, we take these 3-carbon pyruvate waste molecules from glycolysis and we send them into the citric acid cycle. So let's look at where that poised us last time. Here we have over on the left-hand side here is one of our 3-carbon pyruvates. Now remember that glycolysis takes place in the cytoplasm. It is not specified to any particular cell organelle. Now the citric acid cycle occurs within the mitochondrion, which is a specialized cell organelle that is a powerhouse of the cell, the producer of a lot of ATP. So this pyruvate over here on the left has to diffuse actively through the membrane, or the outer membrane of the mitochondrion and inside. So remember now, pyruvate has 3 carbons within it. Immediately, one of these carbons is lost as carbon dioxide. So we've lost one of our 3 carbons. In so doing, blowing off CO2 allows us to reduce an NAD to an NADH. Remember now, in glycolysis, NAD to NADH was using up our NAD, and that was not a good thing. In the citric acid cycle, the formation of NADH is great because that is eventually going to be used to drive the formation of ATP. So we've changed our paradigm here a little bit. Anytime we make NADH, that is a good thing because that is like an ATP IOU. Now that we've blown off 1 carbon as carbon dioxide from our pyruvate, so we have our 2-carbon acetyl group left over here on the right-hand side. This C double bond O CH3, this 2-carbon group, and that is the 2 carbons that are going to get thrown into the citric acid cycle. It is covalently bound to a shuttle, which we looked at the structure of last time, a very large shuttle known as coenzyme A through a thioester bond. And at this point, we're going to look at how this 2-carbon acetyl group gets put into the citric acid cycle. So just to prepare you, this is eventually going to be a very busy slide showing a lot of reactions at once. Now this is just an overview of the citric acid cycle, and we're going to eventually go through each of these reactions to show the structural change from reactant to product, the type of enzyme perhaps or the class of enzyme that might mediate the reaction and things like this. Ultimately, what we have over here shown right now is our acetyl-CoA, and we have that right here, that 2-carbon acetyl group attached to our shuttle, coenzyme A. That 2-carbon acetyl group is then given up by the coenzyme A, and off coenzyme A goes here. And that 2-carbon acetyl group is attached covalently to a 4-carbon molecule known as oxaloacetate. Oxaloacetate. So really what we're doing is we're following the number of carbons here. Oxaloacetate has 4 carbons, 1, 2, 3, 4, and the acetyl group has 2 carbons, 1, 2. That's going, once covalently bound together, obviously, that's going to come up with a 6-carbon molecule known as citrate. So here is a 6-carbon citrate. Notice that ATE on it, like carboxylate. So here, all of our carboxylic acids on citrate are as their carboxylates. If they had if they was not at physiological pH and these were all carboxylic acids, citrate would be known as citric acid. And this is one of why this cycle has the name the citric acid cycle, because oxaloacetate picks up two carbon acetyl group in order to become our citrate or citric acid. So let's go from there, and what I'm going to do now is I'm going to bring in the rest of these reactions into the citric acid cycle. We're going to see this is indeed a cycle, because the starting point, indeed oxaloacetate, which we see over on the left-hand side here, oxaloacetate will, after it goes through this cycle, become oxaloacetate again. In order to do that, our 6-carbon citrate here has to go all the way around and lose 2 carbons. Lose those 2 carbons in that acetyl group to become, again, oxaloacetate. So those 2 carbons we're going to find are all lost as carbon dioxide. So that ultimately that 3-carbon pyruvate is completely catabolized to 3 carbon dioxide molecules. And at the end of all this, we reform that 4-carbon oxaloacetate molecule. So let's see how this works. And we see here that it is indeed a cycle. Citrate, that 6-carbon citrate has to maneuver itself using an isomerase enzyme to become isocitrate. That is still 6 carbons. Following this, isocitrate, 6-carbon isocitrate blows off, look at this, 1 molecule of carbon dioxide, and conveniently reducing 1 NAD to NADH. That's a thumbs up. That's good, because that's going to be cashed in later for ATP. So if we lose 1 carbon from 6-carbon isocitrate, we end up with a 5-carbon molecule, and that is called alpha-ketoglutarate. Alpha-ketoglutarate, and we can indeed count 5 carbons on this molecule. So we've lost 1 carbon as carbon dioxide to form alpha-ketoglutarate. The next step, again, we lose 1 molecule of CO2. That is the last carbon. The last carbon in that original 3-carbon pyruvate molecule. Now it's gone. That entire pyruvate molecule has, in a net sense, entirely been catabolized to carbon dioxide. That blowing off of carbon dioxide, again, drives the reduction of 1 NAD to NADH. So now we have 2 of those in the bank, if you will, which is ultimately a good thing. Alpha-ketoglutarate, after losing carbon dioxide, becomes a 4-carbon molecule right here. You'll notice that, once we lose this last carbon dioxide, this 4-carbon molecule is hooked up again covalently to acetyl-CoA. And we have this molecule right here, which we see as succinyl-CoA. Succinyl-CoA. So again, 4 carbons. Now we have the rest of the citric acid cycle. Now that we've used up all of the carbons in that acetyl group, that 2-carbon acetyl group that was dumped into the citric acid cycle, the rest of this cycle is going to be reforming oxaloacetate. And we're going to take a look at how that works. So let's move forward here. Succinyl-CoA then kicks off its coenzyme A to become simply 4-carbon succinate. Here's coenzyme A down here, that's gone. And what that does is it drives the only actual formation of ATP in this entire cycle. When ADP plus a phosphate goes to ATP. The citric acid cycle, for all its talk about making a whole lot of ATP, is really ATP poor when it comes to actual synthesis of ATP. Most of the ATP derived from the citric acid cycle comes from these ATP equivalents, or IOUs, if you will, the NADH, and as we'll see, FADH2 formation. Hardly any ATP is made directly, just in this one step. So in one turn of the citric acid cycle, we're going to find that only one ATP molecule is physically built. Fair enough. So let's move along. We're down at the bottom of the citric acid cycle now with succinate, and now we're going to form the next molecule known as fumarate. Fumarate, basically we've taken a carbon-carbon single bond and we formed a carbon-carbon double bond. In so doing, we have FAD, our other electron carrier, being reduced to FADH2. That is again an ATP IOU. That is money in the bank, and we're going to use all of these reduced electron carriers in another process to drive the formation of ATP. At this point, they're in the form of IOUs, or ATP equivalents, if you like, but ultimately they will be forming our ATP in general. So let's follow that through. We're at fumarate. And in this case, the double bond on fumarate picks up a water molecule, and water distributes over it to form an alcohol. This gives a molecule known as malate. Malate then converts that alcohol to a carbonyl group to reform our oxaloacetate. And in so doing, we drive the formation of NADH from NAD, again, an ATP equivalent. Importantly, now that we have reformed oxaloacetate, we see that this is indeed a cyclic process, and this reformed net oxaloacetate can pick up yet another acetyl group from acetyl-CoA and continue this process, this cycle, around and around and around, over and over again, because it is indeed a cyclic process. Okay, as promised, we are going to look at each of these reactions of the citric acid cycle in more detail to perhaps tease out some more information about exactly what is going on. Hopefully we'll remember that first process was oxaloacetate over here on the left-hand side. That 4-carbon structure picks up the 2-carbon acetyl group in acetyl-CoA here to form our 6-carbon citrate, or if you like, the citric acid. So that is our starting point. We've handed off our acetyl group to oxaloacetate to become 6-carbon citrate. This citrate, again, now where that citrate is over on the left-hand side in the reactant profile, and we'll look towards the product. This citrate now undergoes an isomerization to form a constitutional isomer. Essentially, what we're doing is we're just moving this hydroxyl group over 1 carbon. We're preparing it for something. You know, if you were to ask, well, look at the enzyme which runs this process, aconitase. What class of enzymes might that apply to? Well, we would think that that would be an isomerase class of enzymes, because we're forming a constitutional isomer going from reactant to product. Next one, isocitrate, then becomes alpha-ketoglutarate. Let's look carefully at what type of reaction we see going on here. Ultimately, this is an oxidation. This alcohol over here on the left becomes this carbonyl over here on the right. Now, this is the basis of catabolism, carbon consistently being taken to a higher oxidation state. Now, why might that be? Why is it important to continually bring the carbon atoms in our fuel molecules to a higher oxidation state? Well, we know that no oxidation can happen in the absence of a reduction. And luckily, if our oxidation is this conversion, well, our reduction is this, and this is what we want to leverage. NAD being reduced to NADH. Why? Because that NADH, when it gets cycled back to NAD, will drive the formation of ATP. So we're looking at these fuel molecules carefully to see, well, how can I oxidize this more? Because if I oxidize the fuel molecule, then I can reduce some of my electron carriers, and that is ATP equivalent. So let's see where we go from our product here, alpha ketoglutarate. We find another oxidation, or what is considered an oxidation. It's not as obvious here. Going from alpha ketoglutarate, we pick up coenzyme A to become succinyl-CoA. This is not as obvious when we're talking about whether this is an oxidation. However, I know it is an oxidation. How do I know that? Because I know that this is a concurrent reduction. Again, NAD to NADH. And both of this reaction and the last reaction drove the formation of NADH, which is a reduction. So the carbon reaction must be an oxidation. I should note that in this particular reaction and the previous one, in both cases, we blew off a carbon in the form of carbon dioxide. So now we are back to a 4-carbon substrate. 4-carbon substrate. And the rest of the citric acid cycle now is taking this 4-carbon substrate and rendering it, really oxidizing it, to become back to oxaloacetate. So let's see how that's done. So now that we have blown off all of what once was our acetyl group, all 3 carbons from that original pyruvate molecule are gone, we are left with this 4-carbon unit, or only 4 carbons that matter, succinyl-CoA. And succinyl-CoA it casts off its covalent bond to coenzyme A, and in the process we form we create the only time that ATP is actually produced directly in one turn of the citric acid cycle. Here over on the left I have an ADP, diphosphate, plus a phosphate, and I am going to convert or bring them together covalently to form an ATP. I have recharged my battery ultimately. So again, while the citric acid cycle is coveted for the high production of ATP, is really ATP poor when you think of directly produced ATP. Virtually all of the ATP produced, as we'll see in the citric acid cycle, is from these ATP equivalents, these reduced electron carriers, NADH and FADH2. But now we have an original 4-carbon substrate back here, and we are going. The rest of the citric acid cycle here, as I said before, is to render that back into oxaloacetate to finish off that cycle. So let's see what happens. Let's see what happens to succinate. Succinate, as we can see here, becomes the molecule fumarate. And what the specific structural difference that happens is, here we have a carbon-carbon single bond, and we go to a carbon-carbon double bond. Now, I am going to submit to you that this is also an oxidation. This is an oxidation going from succinate to fumarate. And the reason why, well, there's 2 reasons why. First of all, we have an FAD here being reduced to FADH2. If that is a reduction, then this must be an oxidation. It has to be, as we've learned before. We can't have a reduction in the absence of an oxidation. Also, let's take a look at the action area here on this reactant molecule. And I would submit to you here, and I think you'd agree with me, that there are 4 CH bonds here in the starting material. Let's look at that same area in the product. And here we have 2 CH bonds. We have lost 2 CH bonds. That is a diagnostic structural change, which ultimately means we have an oxidation. So that is yet another reason that we can call this an oxidation. So now we have our fumarate over here, and let's see what happens to this double bond on fumarate on the next reaction. In this case, fumarate takes a molecule of water over here, and it adds over that carbon-carbon double bond to form an alcohol. Remember that reaction that we looked way back in organic chemistry, an alkene. Water will add, it's an addition reaction, will add over it to form our alcohol. This is not an oxidation or a reduction. It is a hydration if you have to think about this. But let's think about fumarase here as an enzyme. What class of enzymes might fumarase belong to? Well, as we know, the lyase class of enzymes catalyzes the addition of water over a carbon-carbon double bond. So fumarase must belong to the class of lyase enzymes. So now over on the right-hand side, we have our 4-carbon malate, and that is positioned, if we look at this last reaction of the citric acid cycle, we have malate converting back to and closing the cycle back to oxaloacetate. Let's look specifically at what's happening here. I have an alcohol functional group here, but it gets converted into a carbonyl group. That is, by definition, we lose CH bonds and we gain CO bonds, an oxidation. We are taking carbon again to that higher oxidation state. This is why we're always bringing carbon to a higher oxidation state, because it drives the formation of the subsequent reduction. In this case, yet another molecule of NAD is reduced to NADH. And in this case, that is great. That is an ATP equivalent. We're saving those up, and eventually we are going to build ATP through the reconversion or reoxidation of NADH back to NAD. However, not in the citric acid cycle. The citric acid cycle is only for producing a lot of these reduced electron carriers. So we just spent a lot of time talking about the structural changes from these carbon substrates as they go through the citric acid cycle, which is fine. This is an organic chemistry course, so I think it's appropriate that we're actually looking at the carbon changes, the structural changes as we proceed from one substrate to another throughout this cycle. However, it is an energy production cycle, and this is, we are talking specifically about energy production. So there are other ways of viewing the citric acid cycle, essentially bringing down the importance of what the actual structure of these carbon substrates are. We know those carbons are, throughout the citric acid cycle, are moving to a higher oxidation state. So we de-emphasize the structure and, more importantly, emphasize the energy or energy equivalents we're getting out of there. And we can start all the way back at pyruvate. It's also known as pyruvic acid. So it's given as pyruvic acid here. And pyruvate or pyruvic acid is given as these 3 beads on a string, these 3 beads right here. That means it's 3 carbons. So ultimately, the number of carbons has been de-emphasized just to show that they are beads on a string. And ultimately, we have 3 carbons, which loses 1 carbon as carbon dioxide to form our 2-carbon acetyl group. And that is connected to coenzyme A. But you see how this is going. 3 beads loses 1 bead. Well, we go to 2 beads. That is of lesser importance, at least in this way of looking at the citric acid cycle now. But of more importance, in this case, we have this NAD, which is being reduced to NADH in bright yellow. And that bright yellow indicates the emphasis of importance on energy production. So ultimately, like we saw before, we have our 2-carbon acetyl group over here and our 4-carbon oxaloacetate. Even the names on this have been de-emphasized, but 2 carbons plus 4 carbons go to our 6-carbon citrate. And that is the extent of how the importance in this case we're putting on the structure of citrate or citric acid. But really what's going on here now is that these 6 beads on a string in citric acid lose sequentially 1 and 2 of their beads, their carbons, as carbon dioxide, driving 1, 2 conversions from NAD to NADH. So already going from pyruvate, we have 3 NAD in the bank, and we're not even halfway through the cycle. So and ultimately, so this is where the high number of ATP and ATP equivalents are derived from, and ultimately we're going to be squeezing every last potential energy drop out of this. And as we move, as we've went through the last 2 reactions, we see that, oh, look, now we're back at 4 carbons again over here around 5 o'clock on this cycle. And in this case, now these 4 carbons are completely rendered or are continuously oxidized back to that oxaloacetate. So that doesn't change in this case, but what we do see are much more of these bright yellow energy equivalents being formed. Here's our single ATP formed directly from the directly in one turn of the citric acid cycle. Here's an FADH2 formed, and here's that final NADH formed, so that we can sort of add these up now. We can get a summation of the energy that is produced, and it's important to recognize here that 1 glucose molecule, remember 1 glucose is 6 carbons, and that produces 2 3-carbon pyruvates. So that 1 glucose molecule triggers 2 turns of the citric acid cycle. That is important because it's easy to sort of think, oh, 1 glucose, 1 turn of the citric acid cycle. It actually promotes 2 turns per 1 6-carbon glucose of the citric acid cycle. That's going to become important when we start adding up all of these. So, well, how many ATP can we expect from a molecule of glucose? And we'll do that calculation presently. But adding all of these up from pyruvate, we produce 1 ATP directly, a total of 4 NADH per 1 turn of the citric acid cycle, and 1 FADH2 per 1 turn of the citric acid cycle. So I've been blithering on endlessly about how NADH and FADH2 can be cashed in, if you will, for ATP. And finally here, we're given the exchange rate, if you will. When we form ATP, when NADH cycles back or reoxidizes to NAD, and we form ATP when FADH2 similarly oxidizes back to FAD, our original one. Now, the actual exchange rate, if you will, 1 NADH going to back to NAD drives the formation of 3 ATP. 1 FAD going to FADH2, I'm sorry, going to FAD drives the formation of 2 ATP for each one that is reoxidized back. So now we have this understanding of what at least the exchange rate is. I want to focus on NADH because NADH is produced in 2 of the energy production pathways that we've looked at, and in 2 different parts of the cell. So let's look at that carefully, and we'll designate this that we have our glycolysis, which happens in the cytosol or the cytoplasm of the cell. That is freely within the cytoplasm, and in glycolysis, NADH is indeed produced. But the NADH produced in the citric acid cycle is formed inside the mitochondrion. So that is different. It's still NADH, but it is formed inside the mitochondrion. So they are considered different. And the reason for that is while NADH cycling back to NAD will always drive the formation of 3 ATP, we find that NADH produced in the cytoplasm or the cytosol is only worth 2 ATP, not 3. Now, and the reason for this is as follows. When NADH is produced in glycolysis, before when we were just looking at glycolysis, that was a drag. It wasn't. It was a waste product. We had to recruit fermentation to cycle it back to NAD so that glycolysis could continue. However, now that we have this other energy production pathway, this aerobic one, the citric acid cycle, NADH produced in glycolysis here in the cytoplasm or the cytosol can be utilized in the citric acid cycle or after the citric acid cycle to drive the formation of 3-ATP. But it has to get into the mitochondrion to do that. So here over on the left-hand side, we have NADH. It has to make it into the mitochondrion in order to cash in for 3-ATP, which it does. However, in order to get through that mitochondrial membrane, costs exactly, you got it, 1 ATP to get in there. It's like a cover charge in a bar or something like that. The NADH inside the mitochondrion, once it gets in, can still be cashed in for 3 ATP. However, we have to pay back the bouncer, if you will, of that 1 ATP that we burned just getting in the door. So net, we have 3 ATP for that NADH, that cytosolic NADH, minus the 1 ATP that it burned just getting in the door of the mitochondrion. So that's ultimately, so we can now specify different types of NADH when we're trying to come up with a rendering of how many ATP are produced from one molecule of glucose. NADH now produced in the mitochondrion is worth a net of 3 ATP. However, cytosolic, this NAD produced in glycolysis, cytosolic NADH is worth 3 ATP, but you have to pay back that 1 just to get in through the door of the mitochondrion. So it's worth 2 net ATP. Let's see how that works out when we're putting together as I said, a rendering of the number of ATP per an entire 6-carbon glucose molecule. Okay, so the energy yield from one glucose molecule, one 6-carbon glucose shown here on the bottom left. Let's take this one at a time. Remember, from glucose, 6-carbon glucose, to 6 carbon dioxide molecules requires 2 energy production pathways. We have to break down glucose first by way of glycolysis into our 2 3-carbon pyruvate molecules. So in glycolysis, if you remember, what was the net per glucose molecule? Was a net gain of 2 ATP by way of substrate level phosphorylation. 2 ATP per 6-carbon glucose molecule. Also, glycolysis per glucose molecule produces 2 NADH, which now can be taken up and ATP can be rendered from it. However, glycolysis occurs in the cytoplasm. And so this is considered cytosolic NADH right here. It has to, in order to get inside the mitochondrion to form 3 ATP per NADH, it has to spend, if you will, one of those ATP. So this cytosolic NADH, if you will, is worth 2 ATP per NADH. So with 2 of those, that's a total of 4 ATP. So what we're doing now is we're starting to add up the ATPs. So a total of 6 ATP now per glucose molecule comes out of glycolysis. From one of our previous slides, we had added up, once these pyruvates go into the citric acid cycle, we had added up all of the NADHs, FADHs, and formed an ATP from that, and that is our next ultimate step, where we take our 2 pyruvates formed in glycolysis and completely oxidize them to 6 molecules of CO2. So important thing to remember here is in the citric acid cycle, we have 2 pyruvates, so each glucose powers 2 turns of the citric acid cycle. 2 of them, and that's what I'm going to be ultimately adding up here. So per turn of the citric acid cycle, we drive the formation of 1 ATP directly. Now, since we're talking about 2 turns of the citric acid cycle, that is 2 ATP produced directly. 1 turn of the citric acid cycle all the way from pyruvate gives us 4 NADH, but we have 2 of them. We have two turns of the citric acid cycle, so that's a total of 8 NADH, I'm sorry, times 3 ATP per NADH, that is 24 ATP. Lastly, 1 turn of the citric acid cycle produces 1 FADH2. If each molecule of glucose powers 2 turns of the citric acid cycle, that's 2 FADH2 times 2 ATP per FADH2, total of 4 ATP. When we add all of these up, we find that per molecule of glucose, animal systems using those 2 energy production pathways can extract a total of 36 ATP per molecule of glucose. Now, to finally understand how we get 3 ATP per NADH and 2 ATP per FADH2, we have to understand how we get to that conversion factor. And to do that, we're going to take a closer look at the structure of the mitochondrion, sort of a crude drawing of that we see here. We find there are different compartments in the mitochondrion. We had said before that we have 2 membranes in a mitochondrion, an outer membrane, which we see here, and that's given all the way around the mitochondrion, but also an inner membrane. That inner membrane was highly folded, remember, so we have this highly folded nature to the inner membrane, and what that does is it allows a lot of surface area on that inner membrane for processes to happen. These processes being the production of ATP. Now, having 2 membranes, we get compartmentalizes us into 2 general areas within the mitochondrion. Within the inner membrane of the mitochondrion, we have what is known as the mitochondrial matrix in blue right here. This is all considered an area known as the mitochondrial matrix. And it is in the mitochondrial matrix that the citric acid cycle happens. So we have our citric acid cycle happening, we have our pyruvates being converted into carbon dioxide and so on. Everything that we just looked at is happening within that matrix. But another compartment in there is between our inner membrane and our outer membrane, and that is the intermembrane space. That is a compartment, a separate compartment. That inner membrane space or intermembrane space is what we see here in tan. So this whole tan business right in here is our intermembrane space. So it is important in a mitochondrion that we have these 2 segregated spaces. Now, floating, if you will, within our inner membrane are a series of proteins known as the electron-transport chain. The electron-transport chain, 2 of which are shown here, but there are too many to count really. One of the reasons is because we can fit a lot of electron transport chains on the very high surface area that we formed from this inner membrane. The more electron transport chains that we can accommodate embedded into this inner membrane, the more ATP we can produce. So it is at the interface of this electron transport chains that we're going to be reoxidizing NADH back to NAD and FADH2 back to FAD. And ultimately, the way this is going to happen is those electrons contained in the hydrides that this NADH and FADH2 contains are going to be injected, if you will, into the electron transport chain. So let's look at how that happens. Now you'll notice there is another structure here this is called ATP synthase, which is an amazing enzyme, which, as you can probably tell, will produce ATP. But that is sort of at the end of the process. We have to power up ATP through a process first. So this electron transport system, or electron transport chain, has this series of electron carriers. They're embedded proteins in this inner mitochondrial membrane. So let me just set up what we're looking at here, because this is kind of a busy slide. We have a membrane here. It's a lipid bilayer. Here's one side of the bilayer. Here's the other. What that very small area, all the way across, really, so this is all a lipid bilayer, what that represents is a small portion of the inner membrane into which our electron transport chain, these proteins, are embedded. And those 4 proteins in the electron transport chain that are important are represented right here, just these blobs that are embedded into that membrane. And notice 3 of the blobs, which we see here, 3 of them transverse all the way through the membrane. They go from one side to the other, and one of them only goes partially part of the way through. Now, so that's going to be ultimately important. And so we'll look over towards the top of the screen here. We have this is the area, that intermembrane space, that compartment that was in tan in the back in the last slide. In here, on this side of the membrane, all in here, is the matrix, the mitochondrial matrix. So that was that blue that we saw in the last slide, that blue area. And it is in this mitochondrial matrix that the citric acid cycle happens, and we create all of these heavy laden or reduced ATP equivalents, NADH and FADH2. So ultimately, what's going to happen is NADH is going to be cycled back to NAD. It is going to be oxidized back. FADH2 is going to be oxidized back to FAD. And we see this happening here. The way this happens is it loses that hydride, that H- on NADH will lose H- to reform NAD. Remember now, in that hydride, H- contains 2 very energetic electrons. So it's not really the hydride that is important here, but the 2 electrons that it contains. Once NADH here, over on the left-hand side, docks with the first protein in the electron transport chain, it injects those 2 energetic electrons within it. Now, these electrons start a journey then. And that journey, if you will, it follows right here. Here we have 2 electrons, and sequentially those 2 electrons will pass through 1, 2, 3 of these transmembrane proteins which are in that inner membrane, each time getting rid of or losing a little bit of their energy to that transmembrane protein. What the energy lost in those electrons does is it powers a pump, and it pumps 2 hydrogen ions from the mitochondrial matrix into the inner membrane space. Every time we have a pair of electrons going through one of these proteins, it pumps 2 hydrogen ions into the intermembrane space. And look what we see, and then these 2 electrons go through the next protein, pumping 2 hydrogen ions into the intermembrane space. And finally, those the electrons give up the last of their energy going through that 3rd transmembrane protein, again pumping 2 hydrogen ions into the intermembrane space. We'll talk about the importance of those hydrogen ions in just a moment. However, what we're going to be looking at now is the fate of those 2 electrons. Having spent all of their energy now, those 2 electrons that were contained in NADH, in this case, hook up with an oxygen atom and 2 hydrogen ions to form a molecule of water. One molecule of water. So this really brings up a couple of things. We have our 2 electrons from NADH, or we'll find FADH2. They end up in a molecule of water at the end of all this. Also, getting back to the original intent of this energy production pathway, this is an aerobic process. Nowhere yet have I said anything about requirement of oxygen. But finally, in this very last step, this is where the requirement of oxygen is. Why we breathe is we have a half an O2, which is an oxygen atom here. Down, if we look down here, one oxygen atom picks up 2 hydrogen ions and these 2 electrons to form a molecule of water. So for all the importance that we hear of, requiring oxygen, this aerobic system, all an oxygen really does, an oxygen atom, is to provide a garbage can for spent electrons. That's it. That is it. And if we don't have that garbage can and oxygen, this is why we breathe, a constant supply of oxygen going into our bodies, are basically just a huge number of garbage cans we need for these spent electrons. If we don't have the oxygen, we don't have these oxygen atoms, and we don't have the ability to shuttle away these spent electrons in this electron transport chain, and we're not able to create ATP energy. Notice we haven't talked about that yet. We haven't created any ATP energy. We've just been pumping hydrogen ions through the inner membrane into the intermembrane space. But we will address that now. So as NADH is kicking off its electrons, and notice those electrons where NADH docks goes through 1, 2, 3 of our intermembrane proteins. FADH2 docks at a different protein in the electron transport chain. It docks its electrons at this second one here. And notice this one does not go all the way through the membrane. Thus, its 2 electrons, as they pass through this first one, cannot pump 2 hydrogen ions into the intermembrane space. It is only when they go through the last 2, that's worth 2 electrons, protons or hydrogen ions, through that intermembrane space. And now we see why FADH2, or we will see why FADH2 produces only 2 ATP, whereas NADH produces 3 ATP. It's all about the number of hydrogen ions that can be pumped from the mitochondrial matrix into that intermembrane space. What we've done is we've created a pressure of sorts, a gradient. We have a lot of hydrogen ions here in the intermembrane space, and it acts as a source of high pressure. And that high pressure of hydrogen ions are then driven in a controlled fashion through that other magnificent enzyme that we see embedded here in that inner membrane called ATP synthase. ATP synthase allows in a controlled fashion, 2 at a time, these hydrogen ions to rush through it. For every 2 hydrogen ions that rush through ATP synthase, we drive the formation of 1 ATP. For every 2. So notice now, for every 2 electrons that are, hydrogen ions that are pumped into the intermembrane space, that is going to be the formation of 1 ATP. So we can actually do a rendering now, some simple math to look at that. So now that we have at all 3 sites, these protons, hydrogen ions are protons, can be pumped from the matrix into the intermembrane space, and look at what we ultimately get here. This high hydrogen ion gradient drives this formation of ATP from ADP and a free phosphate through this ATP synthase. We can now actually count how each NADH provides 3 ATP molecules. Why? Because the electrons in each NADH push 2, 4, 6 hydrogen ions through. If there's 6 hydrogen ions through in the intermembrane space, and it takes 2 to run through ATP synthase to produce 1 ATP, we find that 3 ATP molecules are the result. Same thing can be said for FADH2 going to FAD. It pushes through, it will push through 2, 4 hydrogen ions. Again, since it takes 2 hydrogen ions to go through ATP synthase to form an ATP, each FADH2 produces 2 ATP molecules. So what follows are just 2 more slides of the exact same thing we looked at, just written a little bit differently. Again, over on the left-hand side, NADH cycling to NAD allows the electrons through 3 of those transmembrane proteins, forming with an oxygen atom water. So that is ultimately, if we, again, follow the bouncing hydrogen ions, that's driving for each transmembrane protein 2 hydrogen ions through these things into that intermembrane space, and these hydrogen ions ultimately make their way over to ATP synthase, where they go through, and each 2 hydrogen ions drives the formation of 1 ATP. Similarly with FAD and FADH2 to FAD, we can only, its electron journey can only hit 2 of those transmembrane proteins, so we get a smaller number of hydrogen ions pumped into that intermembrane space, and thus less ATP produced per FADH2 when we ultimately take a tally of things. Yet another one is really the same sort of thing. Here we are showing both sides now, the mitochondrial matrix and the intermembrane space. Here's where the citric acid cycle happens within that mitochondrial matrix, producing all of those NADH and FADH2, which in both cases here are shown docking at their particular points in the electron transport chain, pushing through, in all cases, hydrogen ions, and those hydrogen ions build up such a pressure that they are forced through, 2 at a time, through ATP synthase, driving the formation of ATP.